I start by thanking the organizers for inviting me to give a keynote in this wonderful virtual session. I'm very glad we can still have it despite all of the upheaval in the world. Today I'm going to tell you about some new work from my lab about a new method that we're developing to actually measure the rates of 3' end cleavage of mRNA molecules. So we're all familiar, of course, with the central dogma of molecular biology, where DNA is transcribed to create RNA molecules, which are then translated at the ribosome to create proteins. Of course, this is an oversimplification of the process where RNA doesn't magically get transcribed from DNA in one step. In reality, that we know that each of these steps are made up of many specific molecular and biochemical mechanisms, where the act of creating an mRNA molecule includes not just transcription, but also mRNA splicing, 3' end cleavage and polyadenylation, nuclear export, localization of the RNA to the proper place, and then a whole host of mechanisms and steps involved in translation. Now, each of these processes have been studied independently in depth for many years. And from that research, we know that they act together to cumulatively establish a steady state transcriptome or proteome, but we actually know quite a bit less about how they're coordinately regulated. And what I'd like to make an argument about is that to be able to really understand how gene expression is regulated, we have to actually start studying the synergy between these individual processes, and that synergy likely happens through a complex interplay of the relative rates of each of these mechanisms. And this premise fascinates me specifically with respect to the diversity of RNA isoforms that are expressed in cells. Now, while tracking the timescales of each of these individual molecular processes, you can imagine that there's a certain amount of time spent in each one, and that the amount of time is regulated by some limiting factor. Changes in the rates or the timescale of any given one step might result in different expression levels or the expression of different relative RNA isoform levels. And this variation in individual molecular events is what we see as steady state variation in mRNA levels. But despite the centrality of the actual kinetics and timescales at which these mechanisms are happening to create the transcriptome, we really have a very poor understanding of, one, how to actually track these rates at a genome-wide level, and two, how they coordinate to give rise to a desired isoform or transcriptome distribution, and ultimately a desired phenotype in any given cell. So the steps of RNA processing, as many of you likely are very familiar with, are thought to be mostly co-transcriptional, such that splicing, for instance, is thought to occur while the rest of the RNA molecule is still being transcribed. And these steps are also thought to be linear in their progression, as they're depicted here. Of course, you first have to transcribe the RNA, but then the sequence of events is usually depicted such that capping happens, then splicing of all the introns, and finally cleavage and release from the chromatin in exactly that order. But there's increasing evidence that the timing and hence the order of these events might actually specifically be regulated and might be altered in specific cellular conditions. For instance, Daniel Larson's lab showed that for the beta globin gene, splicing of a given intron can occur stochastically either before or after cleavage and release of the molecule from pole 2. And when they looked at cells with a mutation in the U2AF core splicing factor, the timing was actually shifted such that splicing occurred entirely after release of the mRNA. Interestingly, this particular mutation in U2AF consistently recurs in cancer cells, potentially suggesting that it may play a broader role in kinetic competition during cancer progression. Another example is from Doug Black's lab, where they profiled RNA after cellular fractionation in macrophages, and saw that sequential waves of gene expression that occurred after lipid A stimulation of the immune system may be gated by the kinetics of splicing and cleavage rather than transcriptional initiation cascades. So these examples and others highlight to me the need to move beyond a steady state characterization of final transcriptome compositions towards a more dynamic understanding of mRNA biogenesis and maturation. So with that, my lab aims to understand the molecular basis for transcriptome diversity, specifically by developing and applying genome-wide methods to measure the kinetics of gene regulatory mechanisms. And one of the first questions that we'd like to tackle is, what are the rate-limiting steps for RNA maturation across different genes? You might imagine that the rate-limiting step in the biogenesis of isoforms from gene A is transcription, and all of the other steps occur relatively rapidly after splice sites or polyadenylation sites become available. However, for other genes, transcription might actually proceed quite rapidly, but the creation of a mature and productive mRNA molecule might instead be gated by the rates of splicing or 3' end cleavage. To tackle that question, the first challenge that we had when I started my lab was to actually develop high-resolution approaches that allow us to systematically and simultaneously measure genome-wide rates of key steps in RNA biogenesis and maturation. And the steps that we focus on are transcription elongation, mRNA splicing, and 3' end cleavage. So I'm not going to talk today about our method for transcription elongation rates, although I'm happy to discuss this offline. Instead, I'm going to start by telling a little bit about a story about how I started this work when I was a postdoc with Chris Burge, measuring the rates of mRNA splicing, and then continue on to tell you about our new work on measuring the rates of 3' end cleavage. Okay, so how do we actually go in and measure the rates at which RNA processing is happening? To do that, we need to be able to capture RNA molecules across a range of time periods, both before and after splicing, for instance. And to do this, we incorporate a metabolic label, specifically 4-thiouridine, and that label is incorporated into newly transcribed RNA in the place of normal uracil bases. And we do that for very short time points, less than 5 minutes, 10 minutes, or 20 minutes. And so the expectation is that with increasing labeling time points, more nascent molecules have the time to be spliced, and therefore you see more intermediates that have not yet been spliced at earlier time points, and that decreases over time. And that rate of intron loss over time allows us to infer a splicing rate, where the half-life for splicing is the time at which half of the transcripts have completed intron excision. And the data I'm going to tell you about today uses a nascent RNA with metabolic labeling with 5 minutes, 10 minutes, and 20-minute incubations for SU in Drosophila melanogaster S2 cells, paired with a steady state data set. And I have to mention that this data was all generated in collaboration with Karen Edelman's lab. Okay, so the way in which I'm drawing this really nice, clean decay in intronic read proportion over time is unfortunately an oversimplification of the data that we actually have, since it assumes that the intron was synthesized exactly around T0, right when the label was actually added. But in reality, we actually have a much more complex distribution of intron lifetimes because our experimental data involves continuous labeling with 4SU over the entire labeling interval, for example, for a full 5 minutes. That means that we're not only sampling introns that initially finished transcription and therefore had the ability to start splicing right at time 0 and had a full 5 minutes to be spliced, but rather we're also sampling introns that were actually created about 4 minutes or maybe even seconds before the end of the labeling period, and therefore only had 30 seconds or maybe a couple seconds to actually have splicing be completed before we extracted the RNA. So to illustrate this in more depth, we can imagine that each of these potential molecules with different lifetimes that I'm drawing on the left here contribute about 8 reads to our final data set. Now, the proportion of exon-exon junction reads, which indicates completed splicing, or intron-exon junction reads, which indicate unspliced molecules, that each of these molecules provide is proportional to both the splicing rate of that given intron and the time that it had to actually splice out the intron. So if all of these reads are coming from the same intron, which has the same aggregate intron half-life, and we knew which reads came from which of these lifetimes, we'd be able to actually easily infer the rate of splicing. However, our data, of course, is an amalgamation of all of these reads across these different lifetimes, making it much more complex to infer the exact lifetime that each read had to be, each unspliced molecule had to be spliced out. Okay, so to account for this complexity of intron lifetimes and their relative contribution to our read pool, we conceptualized this a little bit differently, and we conceptualized it in terms of the distribution of polymerase positions at the end of labeling periods, where each of these cases that I showed you before would fall into one of 3 categories. First, polymerases that are still within actively transcribing introns, and therefore reads in the intron would indicate a molecule that had not yet even started the ability to be spliced. Second, from polymerases where the polymerase was within the transcript, but downstream of the actual 3' splice site, the splicing could have happened at any given time. And finally, from polymerases that finish transcription of the transcript, where the hypothetical distribution of polymerases, in this case, is dependent on both the labeling time and the transcription rate to account for the time after the transcription completed for that transcript. Okay, so we can use this conceptual framework to think about the probability distributions of observing either unspliced transcripts in pink here or spliced transcripts in blue here across these polymerases. And both of these distributions are dependent on the rate or half-life of splicing for the given intron we're interested in studying. Empirically, the only reads that confidently differentiate between unspliced or spliced introns are these junction reads, either exon-exon junction reads or intron-exon junction reads. And so what we did was we derived a model that relates the ratio of these unspliced to spliced reads to these probability distributions. And we used this to solve for the half-life for each intron for which we had sufficient reads to estimate this ratio. So this data in this model is actually already published, so I'm not going to go into too much depth more mathematically or into the actual results. I encourage you to read the paper. But I want to mention that we found a median half-life of intronic splicing in fly cells of a little bit less than 2 minutes, with a huge range of variability. And one of the main things that we were interested in is the idea that introns from the same gene tended to have more similar half-lives or rates of splicing than random introns from many different genes. And this suggested there might also be coordination, not just between the splicing of different introns within the same gene, but also other processes occurring for that particular mRNA molecule. So now I'm going to focus on the unpublished work from my lab, where we extend this method to measure the rates of 3' end cleavage. And this is work by a graduate student, Leslie Torresalua, who realized that we can use nascent RNA sequencing reads and adapt that previous splicing rate model to estimate the genome-wide rates of 3' end cleavage from an actively elongating polymerase. So why do we care about the rate of 3' end mRNA cleavage and polyadenylation? I hopefully don't really have to convince this crowd that the positioning and the efficiency of choosing a poly A site has many consequences for the actual regulation of a given mRNA molecule, since we know that 3' UTRs and therefore the positioning, the choice of a different alternative polyadenylation site can influence many processes such as mRNA stability, mRNA translation, nuclear export, localization, protein localization, and translational impacts, and so on and so forth. So it's been hypothesized that differential cleavage rates across sites in the same gene might actually influence the ultimate isoform usage and expression. One hypothesis has been that the rate of transcription elongation might actually affect which poly A site is around for longer and therefore is more preferentially used. And in this model, the idea would be that a slower polymerase might preferentially lead to usage of an upstream poly A site, while a faster polymerase might allow more usage of a downstream poly A site. However, this model relies, kind of misses one aspect of it, and that is that there's an interplay between both the elongation rate of a given polymerase molecule and also the rate at which each individual site is actually cleaved up at, where the rate of cleavage at an individual site can also impact the given isoform in 3' UTR isoform that's used. Okay. So the first challenge for actually estimating rates of cleavage was to actually identify reads that differentiate between cleaved and uncleaved transcripts, where we realized that we had 4 classes of molecules. First, molecules that haven't yet reached the cleavage site and therefore could not yet be cleaved. Second, molecules that have transcribed past the cleavage site and therefore are either mature mRNA molecules or read-through transcripts that are molecules past the cleavage site after cleavage has actually occurred. And finally, molecules that have transcribed past the cleavage site but not yet been cleaved. So initially we were wondering whether or not we could just use the proportion of reads that occur past the cleavage site and therefore are not protected by poly A tails to estimate cleaved versus uncleaved molecules. But we realized we were competing against the actual degradation rates of these read-through transcripts, where XRN2 quickly starts degrading these read-through transcripts from the 5' end. And so actually estimating any cleavage rate by using reads coming from these downstream molecules would be completely confounded with the rate of XRN2 as well. So we were left with a lack of a clear understanding of any read patterns that we could take advantage of. And so we decided to in silico simulate reads coming from all of these types of molecules and given rates of cleavage to understand better what the spatial distribution of reads are in nascent RNA sequencing data before and after cleavage. So as I mentioned, we simulated reads, and to do this, we first simulated transcripts across a range from nascent RNA sequencing data across a range of different gene lengths, read-through regions, and expression levels, and given numbers of transcripts and reads. So what I'm showing you here is from 3 different labeling time points, molecules that we simulated, where each row here is one mRNA molecule. The dotted line here is the cleavage site, so all of the region before this dotted line is the gene body, and after this region is the read-through region. And the reason for this slope here is because this is nascent RNA sequencing data. These fraction of molecules up here represent molecules that have not yet transcribed past the cleavage site. And then for every molecule that's transcribed past the cleavage site with a certain probability that's dependent on the actual half-life of cleavage, these molecules can either be not yet cleaved, which is in black here, or cleaved, and therefore you have a cleaved mature mRNA and a cleaved read-through molecule that's not a mature mRNA molecule. Okay, so once we simulated these molecules, we started actually simulating the library preparation process. To do that, we first did random fragmentation using a Weibull distribution, as we've described previously. And then we performed a size selection step with a given mean insert size and a given standard deviation around that mean insert size. And one thing you start noticing very quickly is the difference between these black fragments and fragments that are derived from molecules that have already been cleaved in purple here. You start to see that there are more fragments from these uncleaved black molecules that are right over the actual cleavage site, but an actual depletion of fragments right over the cleavage site from these cleaved molecules. And this becomes even more prominent when we actually just look at the paired end reads at 50 nucleotides on either side of these fragments. And so what we realize this represents, and what I'm showing you now, is the metagene plot across all of the molecules that I just showed you, plotted for all of the different labeling time periods, and this very prominent depletion in reads from these cleaved molecules right around the actual cleavage site. And what we realize this represents is an RNA sequencing edge effect, where there's a known depletion due to fragmentation and size selection procedures of reads coming from the very ends of molecules. So for uncleaved RNAs at the bottom here that had not yet been cleaved, there is no depletion of reads right across that region. But from cleaved RNAs, you actually see a depletion of reads because there's now 2 molecules, and these 2 molecules each have an edge effect where the edge is located right at the cleavage site. So this led us to understand that we could actually use the extent of this depletion as information about what proportion of molecules in our data set had actually been cleaved or not at a given labeling time period. Furthermore, we realized that the extent of this depletion was also changing across time points. And so this is heuristic data here, where I'm showing you from 5 minutes, 10 minutes, 20 minutes, and a steady state RNA sequencing data set, that this depletion increases with increasing labeling time periods, indicative of increased proportion of cleaved molecules. So up until now, I've only shown you simulated or heuristic data. So what does this look like in the actual nascent RNA sequencing data set that we have from flies? So here's a metagene plot across 11,000 annotated transcription end sites with the same labeling time points that I told you about for our mRNA splicing measurements, where we have labeling with 4SU for 5 minutes, 10 minutes, 20 minutes, and a steady state total RNA sequencing data set. And you see exactly what I described before. Not only do you see a depletion of reads right around the transcription end site, which is in 0 here, you also see a difference in this depletion across time points. And you see that this depletion starts to normalize out right around the actual size of the fragmentation length, which makes sense given that the fragmentation, the size of fragments and inserts that are selected from that is what actually determines the extent to which you have a depletion of reads in these regions. Furthermore, one last thing that you see is that you have a lower amount of reads deriving from this read-through region to the right of the cleavage site here. And the reason you have a depletion of those reads derived from downstream of the cleavage site is because XRN2 has begun to do its work and is starting to degrade these read-through regions, while the mature mRNA molecules have presumably been polyadenylated and are stabilized. So I showed you a metagene plot. What does this look like around individual transcription ends? I'm showing you two representative examples here, which are noisier, but you still very much see the same pattern across the different time points. So what we realized we could do, given all of these simulations and understanding of the spatial distribution of reads around cleavage sites and nascent RNA sequencing data, we realized we could adapt our previous mRNA sequencing, mRNA splicing rate model to measure the rates of 3' end cleavage. And we make three, I'm not going to go through all of the math and how we do it, but I want to mention three specific aspects that differentiate this model from the splicing rate model. First, we assume that cleavage can only begin after the transcript has actually elongated past the actual cleavage site, and thus beginning at that site, we have an exponential decrease in the probability of observing uncleaved reads, which is in pink here, and an exponential increase in the probability of observing cleaved reads. And both of these distributions are dependent on the actual half-life of cleavage. Second, to differentiate between these two fractions, we use reads that directly overlap the cleavage site to measure the proportion of uncleaved transcripts, and reads from a region that is at least one fragment length upstream of the cleavage site, and that approximates the total number of transcripts in our data, and corresponds to that at the end of that cleavage dependent dip, as we saw in the spatial distribution of our reads. And finally, we make an assumption that we can get informative reads from polymerases that have transcribed any distance from the cleavage site all the way down to the end of the read-through region downstream of the cleavage site. And we empirically determine the length of that read-through region for every single polyadenylation site that we measure at. So finally, I want to show you that this model that we derived may actually accurately estimate the rates of 3' end cleavage. So we extended that simulation across many different insert sizes, standard deviations, read-through region lengths, labeling time periods, and various different expression levels, and of course, across many different putative cleavage half-lives. And with that simulated data, we applied our model to that simulated data, and we estimated the rates of cleavage on these data. And what I'm showing you here is on the x-axis, the actual known half-life that we simulated, and on the y-axis, the half-life that we estimate from our model. And we can see a very nice concordance between our expected half-life and the half-life that our model actually estimates. So armed with this fairly accurate ability to estimate the rate of pre-mRNA 3' end cleavage, we actually applied this to the same metabolic labeling data set in Drosophila across 5 minutes, 10 minutes, and 20 minutes of 4SU labeling. What I'm showing you here is the number of uncleaved reads, the number of gene body reads, which very nicely goes down across time, as we'd expect with increased time for cleavage to occur. We calculated half-lives for a little less than 8,000 annotated transcription end sites in about 6,000 Drosophila melanogaster genes that were expressed in S2 cells. And we find a median half-life of about 40 seconds. So while this median is quite a bit shorter than the median splicing half-life, we do actually see a large range of variability in the half-lives from different polyA sites. And so we, of course, now want to understand what actually underlies the variability in cleavage rates. We know from decades of work on polyadenylation sites that the polyadenylation site itself is not the only thing that actually influences the ability to actually cleave at that site. Rather, there are many RNA binding factors, other sequence elements, and connections to both transcriptional and RNA processing mechanisms that may actually influence both the choice of that site and also potentially the rate of cleavage at a particular polyadenylation site. So we first asked, does the position or sequence of a polyadenylation site affect the rate of cleavage? And when we drill down into the actual sequence elements that might regulate the recognition and cleavage at a given polyadenylation site, we find that not only is the actual polyadenylation site important, but there are also many other sequences that are both upstream and downstream of the polyadenylation site that serve as both locations for other factors to bind onto, and also as other key signaling sequences that might affect the efficiency of cleavage. So each of these sequences has a different location that's thought to be localized around the actual key polyadenylation site. So polyadenylation sites themselves tend to be located about 10 to 30 nucleotides upstream of the actual site of cleavage, while many of these other sequences are located either upstream, such as this very short UGUA sequence, or downstream, such as GUGU sequence of the actual site of cleavage. So the first thing that we did was we went into our transcription end sites, and we actually found the location of the most likely polyadenylation signal that is assigned to that given site of cleavage. And what I'm showing you here is that for 11 canonical polyadenylation sequences that are known to be used in the Drosophila genome, the most likely polyadenylation signal and the frequency of those signals in a region 200 nucleotides upstream of our cleavage site. And what we can see is that the two most predominant polyadenylation signals, AAU, AAA, and AUU, AAA, are also the two most predominant polyadenylation signals that we find upstream of our cleavage sites. And we find that the vast majority of the polyadenylation signals sequences that we located are indeed located between 10 to 30 nucleotides upstream of our site of cleavage. So then we asked initially, does the actual position of this polyadenylation sequence have any relationship to the actual half-life of cleavage at a given cleavage site? And we find that there's no significant correlation between the local location of the actual polyadenylation signal and the efficiency of cleavage. However, when we look for enrichment of short 6-mers in the fastest cleaved transcription end sites, we see that there is a significant enrichment for AAU, AAA, which is one of the most commonly used polyadenylation sequences in the upstream region, right before our fastest cleaved transcription end sites. And we also see this short UGUA signal as well as being significantly enriched. Lastly, we asked, maybe it's not the sequence, but maybe it's the prevalence of these sites that might actually contribute to the efficiency of cleavage. And what I'm showing you here is the distribution of cleavage half-lives on the x-axis for different transcription end sites that have either no evidence for a known polyadenylation signal upstream of these sites, just 1 to 2 K-mers present, 3 to 4 K-mers present, 5 to 10 K-mers present. And what we can see is that with more K-mers, polyadenylation K-mers being present, we see a significant trend towards faster rates of 3' cleavage at those particular regions. So the last thing we wanted to understand is whether or not cleavage at alternative polyadenylation sites could occur at different rates, going back to that hypothesis that perhaps 3' UTR isoform usage might actually be driven by the efficiency of cleavage at a given site, not just other factors influencing usage of that site. So we asked for sites, we grouped all of our transcription end sites into sites that were singletons in a gene, occurred across alternative last exons, where the entire exon is different depending on the polyadenylation site that's used, or tandem 3' UTRs, where there are different polyadenylation sites in this given last exon, and the usage of different sites influences the length of that last exon. And what we observe is that genes with transcription end sites that are singletons in their mRNA molecule tend to have the fastest cleavage rates, where there are multiple polyadenylation sites in the same last exon are slightly slower. And when you are actually making a decision between both alternative polyadenylation and potentially alternative splicing, those are the slowest cleavage sites to be used. We drill down in this even further. We find that not only is there a difference between alternative last exon versus tandem 3' UTR cleavage rates, we also see that the actual positioning of either the proximal or distal poly-A site also influences the rate of cleavage. For both alternative last exons and tandem 3' UTRs, the proximal most poly-A site tends to be cleaved slower than the distal most poly-A site in the given mRNA molecule or in that given last exon. We think this is very interesting because it suggests that maybe there's actually regulation occurring at the temporal scale to regulate the usage between proximal or distal sites, and deserves quite a bit more drilling into both how is this occurring in tandem with the splicing event that occurs for alternative last exons, and also in tandem with the different polyadenylation factors that are known to contribute to the usage of different proximal versus distal poly-A sites. And with that, I'd like to summarize by saying that I showed to you today a robust computational model to estimate the rates of pre-mRNA 3' cleavage. We find that 3' cleavage rates are fast, but still quite variable. And we see that gene distal alternative polyadenylation sites are cleaved faster than proximal alternative polyadenylation sites. We still have quite a bit of work to do. We're actively working on further characterizing and validating many of the factors that actually contribute to the variability in cleavage rates, including aspects like the actual rate of cleavage and polyadenylation usage in steady state mRNA data, the efficiency of splicing for introns upstream of polyadenylation sites, splice site usage, RNA binding protein binding, microRNA binding, and many other such factors. And finally, we're also starting to work on methods to both identify precise sites of cleavage using 4SU data, and also the rates of XRN2 cleavage, which, as I mentioned before, may have confounded our results, but we think we might actually be able to get some very interesting biological understanding of the rate of degradation of read-through transcripts by directly studying this. And so with that, I'd like to thank everybody in my group, especially Leslie, who worked on actually really developing this model and the simulations, our funding sources, and I want to put in a plug for the fact that we are actively looking for postdocs, either experimental, computational, or people with experience in both, to come join us in Worcester, Massachusetts. And I'll finish by just leaving up an ad for a postdoc here, and I'm happy to take any questions.
Great. Thank you, Athma. Thank you very much. That was wonderful. We have a few questions that sort of popped up throughout the talk. And the very first one is the simulated data for precision relative to TES is symmetric in the dip area. The real data is not. What is the reason?
Yeah, it's because we didn't actually simulate the rate of XRN2 cleavage for the downstream region after the dip in cleavage, right around in read coverage, right around the cleavage site. So that's actually one of the things that, as I mentioned, we're going back and fixing in our simulations to allow us to actually develop a method to estimate the rates of XRN2 cleavage. But that's why the real data actually shows that decrease in read coverage downstream of the cleavage site.
Okay. And do you assume a constant rate of cleavage? That was a question before. If so, why would that be constant while the other processes are highly variable in their rates? So this is the question when you develop the model.
Yeah. So so we are aiming to estimate the aggregate rate of cleavage across all of the molecules from the millions of cells that we have short read data from. So we do specifically aim to measure the aggregate rate and not the variability in rates across individual molecules, because I don't think we have the resolution to do that with short read data. One thing that we do assume is, and maybe this might be a little bit of a part of your question, but we have to assume a constant rate of transcription elongation because we don't yet have a method to measure transcription elongation at a very specific localized, for specific localized fashion. That's something we're actively working on, and I hope would actually update our understanding not just of the dynamics of transcription, but also how there's interplay between the dynamics of all of these different processes.
Okay. Another question is, do you think that the understanding of the variations in splicing and cleavage dynamics might shed light on origins of some sequencing noise and artifacts in other RNA-seq experiments?
That's an interesting question. I haven't really specifically thought about it in that fashion. I think from a biological perspective, one thing that we're interested in is, and it has been brought up previously, especially in some of the long read sessions today, it's this idea that you might actually have a lot of noisy splicing, and that might be reflected in a lot of the data sets that we look at, even steady state data sets. And I think that understanding the dynamics of splicing and cleavage might allow us to understand not only how much time there is to make a mistake in making a choice of a given splice site or a given cleavage site, and therefore get a non-canonical site usage, or in some understanding of how fast those noisy or non-productive transcripts might get degraded. I haven't really thought about how our understanding of these biological processes might inform technical artifacts, but I will certainly try to think about that further.
Okay, we have another question that sort of asks about conservation. So how is the conservation of these polyadenylation sequences and its impact on processing among other species? So have you correlated your kinetic data with conservation?
We haven't yet, but that's a great idea. Yeah, that's definitely on our list of things to do as we're finalizing some of these analyses.
And then we have another one that's very cool talk. For your quickly cleaved pass motif enrichment analysis, were you exclusively looking for canonical pass sequences, or were you also looking for other motifs too?
I think you're talking about this slide. You can still see my slides, where we were looking for 6-mers. This was completely unbiased. We did not specifically look for polyadenylation sites. We looked for any 6-mer that was enriched, but we did look only in the 200 nucleotides upstream of the cleavage site. For this right plot, we only looked at known polyadenylation K-mers. So these are kind of both sides of the unbiased and biased question, I think, that you're addressing.
I had a question, and that refers to, I remember there was this plot where you plotted a polyadenylation signal distance from the cleavage site. I mean, there were some data points that were, yeah, that one, hundreds of nucleotides away. So, I mean, you understand that that's impossible, right? Yep. Because, so if for those guys, if you go into those, what do you think is driving polyadenylation?
That's a really great question. I don't know. So I should elaborate a tiny bit further on how we actually made this plot, just for clarity. What we did was we looked for evidence of any of these 11 6-mers that have been published before as the kind of most commonly used. It's also been thought that these two in purple here are used about 90% of the time in most eukaryotic species. So if in anywhere in these 200 nucleotides, one of these sequences was present, we assigned that as the actual cleavage site. And if neither of these sequences were present, then we moved to the next closest to the cleavage site, the next one of these green 6-mers. So for these cases here, there really was none of these 11-mers located upstream of this. So we don't necessarily think that this is an error in the actual location of cleavage, because all of the transcription end sites that we're using have been validated with 3p-seq data in Drosophila S2 cells. So I don't really know what could be going on in basically this entire range here from 100 to 200 nucleotides upstream of the cleavage site, but we'll definitely start looking further into those. And
maybe the very last one is So it is known, at least in human, that often when you, I guess you're referring this to tandem passes. This is,
Right.
you know, within the same UTR, you have two alternative passes. That the upstream one is typically the weaker one.
That's right.
So that goes hand in hand with your rate evaluations, right?
That's right. Yeah. Yes, certainly. So I think that might be part of the story as to why here we're actually seeing that the more proximal or the upstream site in both the alternative last exons and in the tandem UTR case tend to be cleaved slower. Interestingly, though, when we look for kind of weaker versus stronger polyadenylation sites only in the single TES category, we don't actually see polyadenylation site strength impacting or a correlation between polyadenylation site strength and the rate of cleavage. So we think it's both, it could potentially be both the strength of the signal, but there might also, there's stronger evidence that there's also a relationship to the actual position and alternative choice that's being made.